When we think of primary productivity, we often think of photosynthesis. However, those of you who have studied marine biology are likely familiar with chemosynthesis. This is where microorganisms oxidize inorganic molecules such as hydrogen, sulfide, ferrous iron, and ammonia, producing water, sulfides, rust, and nitrate. Arguably, the most iconic example of chemolithotrophy are the bacteria which inhabit deep-sea black smokers, which oxidize iron and sulfur released by the vent. But when organic matter is broken down and the carbon is oxidized by heterotrophs, in any environment, the nitrogen and sulfur once bound in the biomass is released as ammonia and sulfide. Nitrifiers and sulfur oxidizers react these chemicals with oxygen, producing nitrate and sulfate to power their metabolism. Furthermore, when organic matter is fermented due to the lack of any electron acceptors, many prokaryotes will produce hydrogen, and when this diffuses into oxic sediments, hydrogen oxidizers will metabolize it for energy. Although chemolithotrophy is a cog in the nutrient cycle in every habitat, recent research in the greening lab at Monash University has indicated that it dominates primary productivity in many more environments than previously realized. More specifically, a version known as trace gas oxidation appears quite common in some of the most desolate habitats on Earth. Even though these habitats will seldom have a source which produces hydrogen, methane, and carbon monoxide, these gases are trace constituents of the air. The concentration of methane in the atmosphere is 1.7 parts per million, hydrogen is 0.5, and carbon monoxide is 0.1 ppm. Chemolithotrophs inhabiting these environments oxidize gases in the atmosphere as a small yet omnipresent source of energy. The metabolism of hydrogen oxidizing organisms, which use oxygen as the electron acceptor, or aerobic hydrogenotrophs, can be thought of as an odd mixture of that of aerobic and photosynthetic organisms. In order to generate energy, hydrogen is oxidized and the electrons flow down the electron transport chain to a cytochrome oxidase. Here, oxygen acts as the terminal electron acceptor. As the electrons flow, their energy is used to pump protons out of the cell, generating a cross-membrane gradient and proton motive force. This is then used to drive ATP synthase to replenish ATP. Some of the electrons taken from the hydrogen molecules reduce NADP to NADPH, which then shuttle them to the cytoplasm. Here, the Calvin-Benson cycle, which is also used by oxygenic photosynthesizers, reduces carbon dioxide using the electrons on NADPH to do so, and it also uses the energy from some of the ATP to assemble this organic carbon into glucose. But how much air would these organisms need to grow and reproduce? The elemental stoichiometry of biomass is depicted here, meaning that on average, organic matter will have 32.4 grams of dry weight per mole of carbon atoms. It will take 2 hydrogen molecules per carbon dioxide molecules assimilated, with one joining an oxygen atom to produce water. Given that there's 9.4 millimoles of air molecules in a liter, it would take the hydrogen in 13,128 cubic meters of air to produce 1 gram of organic carbon. However, simply producing organic molecules from carbon dioxide isn't enough to grow and reproduce. The organic molecules have to be assembled into complex biomolecules, a process that requires energy. How much energy? Well, chemolithotrophic acetogens, which oxidize hydrogen using carbon as the electron acceptor to produce both biomass and energy, only assimilate 10% of the carbon that they uptake as biomass, releasing the rest as acetate, i.e., only metabolizing it to meet their energetic requirements. With 105 kilojoules being generated for every mole of acetate produced, it would take 525 kilojoules per mole of organic carbon converted into biomass. With the aerobic oxidation of hydrogen producing 286 kilojoules per mole, it would take 1.8 moles of hydrogen per mole of organic carbon that was turned into biomass. We can round this off to 2 because a trace gas oxidizer would likely have to do things an acetogen would not, like reduce other biophilic elements. So, roughly half of the hydrogen metabolized would be used to reduce carbon, while the other half would be used to meet the energy requirements of the cell. This means that at minimum, it would take 26,256 cubic meters of air for every gram of biomass produced. Instead of looking at the total biomass, we can look at the number of cells. The average dry weight of an E. coli cell is 3 times 10 to the minus 13th grams. So, 1 liter of air would theoretically have enough hydrogen to produce 126,960 cells. Two other trace gases which can be used by microorganisms are methane and carbon monoxide. The oxidation of methane is initiated by the methane monooxygenase enzyme, which produces methanol. The methanol is then further oxidized to formaldehyde by the methanol dehydrogenase enzyme, which has quinones in the membrane acting as the electron acceptor. Since formaldehyde has a similar elemental stoichiometry to biomass, i.e. CnH2nOn, at this point, it can either be further oxidized to generate energy or assimilated into the biomass of the organism. If the former is the case, it will be bound to the H4-MPT cofactor and oxidized into formate by a number of enzymes until finally being oxidized to carbon dioxide using NAD as the electron acceptor by the formate dehydrogenase enzyme. If instead it's assimilated, most methanotrophs use the RUMP pathway. Here, 3 5-carbon ribulose 5-phosphate molecules will react with 3 formaldehydes to produce 3 6-carbon hexylose 6-phosphate molecules. 2 of these are broken down into 3-carbon glyceraldehyde 3-phosphates, consuming 2 ATP molecules. One of these is converted into pyruvate and fed into biosynthetic pathways, while the other 3 are reacted with the hexylose 6-phosphate to regenerate 3 ribulose 5-phosphates for the next turn of the cycle. Under the technical definition, methanotrophic microorganisms are heterotrophic since methane is an organic molecule. However, since most other heterotrophs metabolize decomposing biomass, they can obtain other biophilic elements such as sulfur, nitrogen, and phosphorus in a form bound to organic molecules. Meanwhile, methanotrophs must obtain it in an inorganic form. Most methanotrophs that have been previously studied live at the interface of oxidizing and reducing environments, and within the reducing environment, much of the inorganic nitrogen and sulfur will be in the form of ammonia and sulfide. Given that these are in the same redox state that they occur in organic matter, they're relatively easy to assimilate. In the oligotrophic environments trace gas oxidizers dominate, it's another story. Here, sulfur will be available in the ground as sulfate-based minerals, and nitrogen may be available as nitrate salts. This means that any trace gas oxidizer would also have to reduce sulfur and nitrogen compounds to obtain the other biophilic elements. If nitrate salts aren't available, nitrogen can be taken from the air as diatomic nitrogen if the organism possesses a nitrogenase enzyme, but this is an energetically expensive process. The last trace gas used is carbon monoxide. This is the least abundant with an atmospheric concentration of 0.1 parts per million. Trace gas oxidizers use the carbon monoxide dehydrogenase enzyme to oxidize carbon monoxide to carbon dioxide. Prior to the discovery of trace gas oxidation, it was thought that methanotrophy, hydrogenotrophy, and carbon monoxide oxidation were carried out by separate microbial OTUs. However, most trace gas oxidizers have mixotrophic metabolisms and can oxidize all 3 of these gases. One hurdle trace gas oxidizers face, which other organisms that metabolize these gases don't, is the low concentrations present in the atmosphere. If you take a mole of an enzyme and plot the concentration of the substrate on the x-axis and the rate at which it's consumed on the y-axis, as you raise the concentration, the rate at which it's consumed will also increase. But The rate will then begin to level off. Over time, the reaction rate will increase at a slower and slower rate as it approaches a horizontal asymptote. This asymptote is known as the Vmax rate, and the reason it slows is because you'll begin to saturate the active sites of all the enzymes. Essentially, virtually all of them will have their active sites occupied, so adding more substrates won't increase the reaction rate. This pattern is known as Michaelis-Menten kinetics. Apart from the Vmax, the other parameter is the half saturation constant, or Ks, the concentration at which the reaction rate will be half the maximum rate. The lower the half saturation constant, the more rapidly the enzyme can use a substrate at low concentrations. How well the enzyme can metabolize substrates at these concentrations is known as its affinity. Usually, if a variety of enzyme has a high maximum rate, it will have a low affinity. So, there will be a positive correlation between Vmax and the half saturation constant. The implication of this is that virtually every environmental microorganism will be faced with a trade-off. If it can metabolize substrates rapidly, it won't be able to utilize the substrate at low concentrations. Conversely, if it can use the substrate at low concentrations, it can only metabolize it slowly at any given concentration. Because trace gas oxidizers metabolize methane, hydrogen, and carbon monoxide at lower concentrations than other microorganisms, their enzymes have to have incredibly high affinities for these gases, and this would also make the max rate at which they can use it very slow, and thus such organisms would grow and divide at very slow rates as well. However, exactly how the enzymes used by trace gas oxidizers are modified to metabolize substrates at such low concentrations remains uncharacterized. One of the major conundrums is that the hydrogen-splitting hydrogenase enzymes are inhibited by the presence of oxygen, suggesting that the hydrogenase enzymes used by trace gas oxidizers have been modified to be aerotolerant, but these modifications again remain undescribed. Trace gas oxidizers dominate primary productivity in some of the most desolate habitats on Earth, including arid desert soils and glaciers. In Antarctic dry soils, hydrogen appears to be the preferred trace gas. Metagenomic analysis of Antarctic samples indicated that trace gas oxidation is carried out by a specialized but diverse assortment of microbial OTUs. This means that individual OTUs which inhabit these environments have become specialized to inhabit such systems, and several different lineages have evolved quasi-independently to live here. I say quasi-independently because while the bacteria which inhabit these environments are phylogenetically diverse, the hydrogenase enzymes used by 9 of these bacterial phyla phylogenetically cluster together. This indicates that the various microbial OTUs acquired the ability to metabolize hydrogen at atmospheric levels by obtaining the gene for the hydrogenase enzymes laterally versus evolving their own enzymes independently. A recent experiment housed microorganisms in chambers with either hydrogen, methane, or carbon monoxide at minus 20 degrees Celsius. While the concentrations of methane and carbon monoxide remained stable, the concentration of hydrogen decreased, seemingly indicating that the cells were still active at freezing temperatures. However, there could still be other explanations for this observation. The hydrogenase enzyme possesses a series of iron-sulfur clusters known as ferredoxins, which facilitate the flow of electrons from the hydrogen to the electron transport chain. Hydrogen molecules themselves are much smaller than methane or carbon monoxide molecules, and could thus more readily move between the water molecules, forming the ice crystal lattice. This would allow them to reach the hydrogenase enzymes in the cell, where the molecules would be oxidized, and the electrons from them will reduce the ferredoxins. But with the rest of the cell frozen, and other carriers like NADH locked in place, the depletion of hydrogen from the gaseous media doesn't necessarily mean that the cells are metabolically active. Another system where trace gas oxidation dominates primary productivity is the subphotic zone of the open ocean. The ocean is supersaturated with hydrogen and carbon monoxide relative to the air, and thus provides an energy source for prokaryotes which don't live in proximity to volcanic vents, yet beneath the photic zone, where little light energy is available. While hydrogen is the preferred trace gas in desert soils and glaciers, carbon monoxide is the preferred substrate in marine systems. This is likely because photochemical reactions between sunlight and dissolved organic carbon will readily produce carbon monoxide molecules. Hydrogenases and carbon monoxide dehydrogenase enzymes were found in proteobacteria, bacteriota, actinobacteria, chloroflexota, Myxococcota, and SAR324, while hydrogenases were present in cyanobacteria, planktomycota, and Aramiobacteriota. I think I should mention that while primary productivity in the subphotic zone will be dominated by trace gas oxidation, the concentration of trace gases will be over 1,000 times lower than that of dissolved organic carbon. So, the oxidation of said organic carbon would still dominate metabolic activity at these depths. While why microorganisms inhabiting the subphotic zone would utilize trace gases is fairly intuitive, why microorganisms in deserts and glaciers would use this strategy is a bit of a conundrum. This is because here, light can provide far more energy than can be obtained by oxidizing trace gases in the atmosphere. The most likely explanation I can think of is that while sunlight can provide large quantities of energy, it is also toxic with significant amounts of UV radiation that kills cells. It is true that photosynthetic microorganisms live on the surface in semi-desert soils. However, many of these form biofilms known as biocrusts on the surface, which include photoprotective pigments for UV shielding. Producing these biocrusts requires many nutrients, and it may be that in ultraligotrophic environments, the microorganisms which inhabit such systems can't produce sufficient biomass to make biocrusts. Now, it is true that chemolithotrophs have been observed in environments directly exposed to sunlight, such as hot springs. The difference here is that there are far greater concentrations of methane, hydrogen, and carbon monoxide. As such, the organisms living in proximity to geothermal sources of gases would have a greater energy budget, allowing them to repair damage caused by solar radiation faster than the cell would incur it. Something resembling the converse of this may have been observed in the most desolate environments on Earth. In the McMurdo Dry Valleys, metagenomic analysis has indicated that there were no microorganisms in some of the soils. One explanation for this is the high concentrations of perchlorates. These are produced by photochemical reactions involving chloride salts and are highly reactive and thus toxic to life. Organisms can survive in environments with perchlorates present, but repairing the damage caused by perchlorates is again energetically expensive. And it may be that living off trace gases doesn't provide enough energy to repair the damage caused by the perchlorates faster than it accumulates in the cells. Apart from meeting the organism's energy needs, it's also hypothesized that trace gas oxidation can help meet the cell's hydration requirements in arid ultraligotrophic environments. At first, this may seem strange, given that the concentration of water vapor in the driest deserts is 200 parts per million, 400 times greater than that of hydrogen. But if an organism was to let water in, it would have to have a less permeable cell membrane, and this would lead to a net loss of water. Since hydrogen molecules are smaller and nonpolar, they would readily penetrate a waterproof barrier. But upon reacting with oxygen to produce water, the resulting molecule would be locked inside the cell, circumventing the permeability dilemma. Going back to our earlier calculations, the reduction of a carbon dioxide molecule to organic carbon will produce one water molecule, while the energy requirements of the cell would oxidize two more hydrogen molecules, producing 3 water molecules per mole of carbon assimilated. The molecular weight of water is 18 grams per mole, and since the dry weight of organic matter is 32.4 grams per mole of carbon, if the cell got all its water from the hydrogen it metabolized to meet its growth and energy requirements, it would be 62.5% water by weight. Most bacterial cells are roughly 80% water by weight, so metabolizing hydrogen at the base rate wouldn't produce enough water. If the cell was to meet its hydration requirements by oxidizing hydrogen, it would have to use 4.2 additional molecules per mole of carbon assimilated, more than doubling the hydrogen it needed. When looking at primary succession after fresh ground is exposed by a retreating glacier, trace gas oxidizers are often the first to colonize the substrate, seeding it with nutrients and allowing other organisms such as lichens to grow on the exposed surface. Although trace gas oxidizers dominate primary productivity in the most desolate environments, such organisms are found across all ecosystems. Furthermore, while many microbes essentially live off trace gases, others facultatively engage in trace gas oxidation. We often hear the term dormancy, or when an organism ceases metabolic activity, but the term is more nebulous than most people realize. The quote-unquote hardest form of dormancy is when microorganisms form an endospore and any metabolic activity ceases. Other forms of dormancy include arresting cellular reproduction and/or growth when metabolism slows to a crawl. Often, this torpor-like state is induced by a lack of external electron donors to power metabolism. Recent research has shown that many microorganisms which use standard heterotrophic processes can oxidize trace gases to maintain their minimal metabolic requirements during the state of quasi-dormancy. This strategy appears to be a common one for microorganisms inhabiting soils. A total of 70% of microbial OTUs in soils have genes involved in trace gas oxidation. This suggests that in these systems, it's largely used facultatively as a means to generate supplemental energy, such as during torpor. The preferred gas in soils is again carbon monoxide, although hydrogen is a much closer second than it is in marine systems. Here, you'll also have photochemical reactions with sunlight and volatile organic molecules producing carbon monoxide. You'll also have much higher rates of microbial fermentation producing hydrogen. However, this does stretch the concept of living off thin air because here, the bacteria would be getting much of the reductant from a localized source of production versus the ambient atmospheric concentration. Apart from ecological significance, trace gas oxidizers can provide insight into how life could survive on the early Earth or on other planets. 20 plus years ago, much emphasis was placed on the roles that reduced gases such as hydrogen, ammonia, methane, and sulfide had on the first organisms. Such attention waned after analysis of ancient zircons indicated that instead of being reducing, the early atmosphere was inert, being mostly composed of nitrogen and carbon dioxide. But the realization that microorganisms can use trace gases in the atmosphere resurfaces questions about the role reduced atmospheric gases had in the ecology of the primordial Earth. Acetogenesis and methanogenesis are likely the oldest assimilatory metabolic processes in existence. Most of the research that looked into how these organisms could have lived on the early Earth has looked into the microbial ecology of geothermal sources of hydrogen, namely volcanic and serpentinizing vents. However, the fact that microorganisms can use atmospheric trace gases raises the possibility that these organisms colonized the open ocean even before photosynthesis evolved. Like the aforementioned acetogens, chemolithotrophic methanogens oxidize hydrogen using carbon dioxide as the electron acceptor. Since carbon is less electronegative than oxygen, less energy will be produced per mole of hydrogen metabolized. But even though hydrogen wasn't a major constituent of the primordial atmosphere, it was still in all likelihood more abundant than it is today. If the hydrogen concentration was greater than 5.5 parts per million versus zero point 0.5 of today, more energy could be attained from a liter of air by an ancient acetogen than an aerobic hydrogenotroph could today, and for a methanogen to do so, it would only have to be greater than 4.3 ppm. It's also true that these early microorganisms lived before the enzymes needed to reduce diatomic nitrogen and sulfate to ammonia and sulfide to be assimilated into their biomass evolved. However, while ammonia and hydrogen sulfide weren't major constituents of the atmosphere, They still would have been minor components and could be obtaken by ancient trace gas oxidizers. Trace gas oxidation has astrobiological significance as well. On Mars, the concentration of hydrogen is roughly 15 parts per million. While this may seem a lot compared to Earth's zero point 0.5 parts per million, Mars's atmosphere has, at its lowest altitude, about 1/80th the pressure of Earth at sea level. So, while the partial pressure of hydrogen in Earth's atmosphere is 51 millipascals, on Mars it's only 17. Could hydrogenase-like enzymes evolve to metabolize hydrogen at these concentrations? Well, without an understanding as to how trace gas oxidizers metabolize hydrogen at atmospheric concentrations, it's difficult to say. With the Martian atmosphere being 95% carbon dioxide, intuition would tell you that the metabolic process consuming hydrogen would be methanogenesis. However, the Martian atmosphere does have oxygen. The concentration is much lower than it is on Earth, 0.16% versus 21%, but it's still 100 times as abundant in the Martian atmosphere as hydrogen is, so any aerobic hydrogenotroph on Mars wouldn't be limited by the availability of oxygen. A more suitable electron donor on Mars would be carbon monoxide. Its concentration in the atmosphere is 700 ppm versus Earth's zero point 0.1. Even when you adjust for the differences in atmospheric density, the partial pressure of carbon monoxide in the Martian atmosphere is roughly 800 millipascals versus 10 on Earth. So any Martian organism with an analog of the carbon monoxide dehydrogenase enzyme would have access to more energy than its Terran counterpart. Although a trace gas oxidizer on Mars would still have to metabolize hydrogen to produce organic molecules. Would the organism react with carbon monoxide or carbon dioxide? Well, each would have benefits. Carbon dioxide is far more abundant in the Martian atmosphere, but carbon monoxide is more reactive and thus would be easier for an enzyme to catalyze. One major hindrance to life on Mars is the high concentrations of perchlorates in the soils, which would present any surface microbes with the same dilemma that those in the McMurdo dry valleys face. This has led many astrobiologists to argue that if there is any life on Mars, it would exist in the deep crust kilometers down, where reduced gases would be emitted by the mantle. Accessing the deep crust likely wouldn't be possible until we build settlements on Mars. However, while atmospheric trace gas oxidizers likely couldn't live on the surface, they would be able to survive a few tens to hundreds of meters below the topsoil, away from the perchlorates, i.e., at a depth a probe could access. Even if the crust of Mars is sterile, between 3.2 and 2 billion years ago, the planet would have been arid, but not hyper-arid as it is today, and instead would have been comparable to the true deserts of Earth. There would have even been ephemeral bodies of water such as streams which were fed by the meltwater from glaciers. Furthermore, sporadic volcanism would release reduced gases into the atmosphere. So, during this time period, Mars would have resembled a mix of the Atacama Desert and Antarctica at a planet-wide scale, potentially hosting a single global biome with primary productivity driven by trace gas oxidation. For more info on the geologic history of Mars, you can see the Life on Mars series. When we imagine life on Europa, we often think of organisms inhabiting a deep-sea hydrothermal vent in the subsurface ocean. These hypothetical organisms would survive by reacting methane, sulfide, and iron from the vent with oxygen in the water column. Where did this oxygen come from? High-energy solar wind particles regularly get caught in Jupiter's magnetic field and then split water molecules in Europa's icy crust, producing hydrogen and oxygen and giving Europa a thin atmosphere of molecular oxygen. Being the lighter element, most of the hydrogen would fly off into space, but some of it would percolate into the crust with the oxygen. So, instead of just living around a volcanic vent at the bottom of the sea, microorganisms on Europa could live in parts of the crust where water seeps through the cracks, oxidizing the hydrogen for energy. Going beyond the familiar reaches of our solar system into the interstellar void, we can find wandering or rogue planets. Most of these would be ice balls frozen solid, but if one of these planets is a gas giant and the moon orbits close enough, it can be heated by tidal forces. This can give the Moon a subsurface ocean similar to that of Europa, and modeling experiments have indicated that an atmosphere composed of hydrogen could induce a sufficient greenhouse effect to allow liquid water to exist on the surface. In order to produce such an atmosphere, the mantle would have to be highly reducing. Yet, local variations in magma chemistry and temperature can produce some volatile oxidized chemicals such as carbon dioxide and sulfur dioxide. This can in turn lead to a biosphere powered by the utilization of trace gases in the atmosphere, but in a way that's the converse of what we see here in the most oligotrophic environments on Earth. Here, trace gases are reduced, using the ample hydrogen in the air as the reductant. Using carbon dioxide or sulfur dioxide as the electron acceptor, microbes could produce methane and hydrogen sulfide. Now, it may be that in such a reducing environment, small-scale chemical variations can't produce the fully oxidized form of volatile elements. But if this is the case, partially oxidized chemicals such as formaldehyde and hydrogen disulfide are more likely to be available than their fully oxidized form, and these can still be used as the electron acceptor. Now, let's venture off further to exoplanets and investigate whether or not the metabolization of atmospheric gases could support a biosphere comparable to that of Earth. In the field of exoastrobiology, little stirs up more controversy than the question of whether or not red dwarf systems could support habitable planets. Due to the proximity of the habitable zone to the parent star, any planet in it would be tidally locked. This has led to arguments about whether or not the atmosphere would persist on such a planet. The most common argument for why there wouldn't be an atmosphere is that it would freeze on the dark side of the planet. Modeling experiments have indicated that circulation would allow the atmosphere to persist, but another contention is that being so close to the parent star, the solar wind would erode the atmosphere. However, being so close to the parent star would also lead to tidal heating, which would in turn lead to higher rates of volcanic activity. While the solar wind would blow the atmospheric gases into space, the outgassing by volcanoes could replenish it. Furthermore, since the lighter hydrogen atoms would be more readily blown off into space than heavier elements, the solar wind would also have an oxidizing effect, even producing oxygen by splitting water molecules high in the atmosphere, in a manner similar to what occurs on Europa. If the mantle was reducing, methane would be released by hydrothermal systems, and could be reacted with abiotically produced oxygen as an energy source. There are reasons to think that in this situation, oxidizing atmospheric gases would be preferable to photosynthesis. For one, half the planet would be in perpetual darkness. Second, the conversion of environmental chemical energy to cellular energy is about 50% efficient, while the conversion of light energy is only 6% efficient. Finally, red dwarfs are flare stars, meaning any photosynthetic organism would have to cope with sudden, intense bursts of UV light. On such a planet, intense wind resulting from the circulation of the atmosphere would be a constant. As such, chemosynthetic organisms could evolve structures to channel the wind toward gill-like organs, allowing them to use the intense airflow to capture as many reduced gases as possible. While it's easy to imagine a sessile multicellular organism surviving by chemosynthesis, as do deep-sea tube worms via microbial symbionts, could active animal-like organisms survive off thin air? Well, we can start by looking at our own physiology. Our basal metabolic rate is around 121 watts. We breathe about 6 liters of air per minute, or 100 milliliters per second, and our lungs can absorb about 20% of the oxygen we inhale per breath. The maximum stable methane concentration, that which won't be explosive, is about 5%. Burning 1 mole of methane will yield 890 kilojoules of energy, and there are 0.0094 moles per liter of air. So, if we could metabolize methane in an atmosphere that was 5% methane, when we crunch the numbers, we could get 8.4 watts, not enough to sustain our metabolism. But when we engage in strenuous exercise, we breathe as much as 100 liters of air per minute. At this rate, the energy per time yield is roughly 139 watts, which is enough to sustain our metabolism. Breathing heavier would require more energy, but we wouldn't be using nearly as much energy digesting food. Also, there are ways to make the lungs more efficient at absorbing gases, such as having the unidirectional system characteristic of birds. Furthermore, the atmosphere could remain stable with methane concentrations greater than 5% if the oxygen concentration is lower. As long as the methane to oxygen ratio is less than 2 to 1, oxygen won't be the limiting reagent. So, animal-like organisms as complex as us could potentially survive off atmospheric gases on exoplanets. One example of this in science fiction might be the grunts from the Halo franchise. These alien adversaries famously walk around with methane tanks on their back that they need to quote-unquote breathe. Their homeworld is known as Balaho, which, according to the lore, has a thick atmosphere composed of methane, carbon dioxide, and helium. Yet, columns of fire supposedly exist there, suggesting that oxygen is also present, which can ignite geothermal sources of methane. So, it may be that grunts inhale methane and oxygen, use the oxygen to oxidize the methane, and exhale carbon dioxide and water. Now, carrying around a large flammable tank of methane would likely be a hindrance in combat, and if grunts use oxygen as the electron acceptor, you'd think surely then they could oxidize their own biomass, as our own cells do. But in order for biomolecules to be fed into the respiratory TCA cycle where they get oxidized to carbon dioxide, they first have to be broken down into simple 2 and 3 carbon molecules such as pyruvate and acetyl-CoA. With atmospheric methane, an omnipresent electron donor on their homeworld, grunts may not have evolved the ability to produce such intermediates and can only power their metabolism by oxidizing methane. You might think that if this was the case, there would be some discrepancy because the grunts often reference consuming food. However, it could be that organic matter which they consume orally is only assimilated into their own biomass, while the methane which they inhale is only oxidized for energy. This brings me to two possible strategies that aliens inhabiting our theoretical atmosphere, which includes methane and abiogenic oxygen, could employ: lithotrophy and organotrophy. The organotrophs would employ the same strategy as grunts, obtaining biomass by consuming organic matter, and in doing so obtaining the other biophilic elements such as nitrogen, phosphorus, sulfur, and iron. Meanwhile, the lithotrophs would obtain biomass by partially oxidizing methane and getting the other biophilic elements in inorganic forms such as ammonia and sulfide. While sulfur, nitrogen, and maybe phosphorus would be available in a gaseous form, the same would not be true of other elements like iron, magnesium, and potassium. So, these lithotrophic animal-like aliens would either have to obtain these by consuming rocks or drinking water with ions dissolved in it. Being able to oxidize all the methane they inhale for energy, the organotrophs could afford higher metabolic rates, but at the expense of having to consume more. This could then lead to a similar niche partitioning that we see between warm-blooded and cold-blooded animals on Earth, with warm-blooded animals being more active at the expense of having to consume more. Well, that's all I have to say on the topic for now, but if you have any contentions or something you'd like to add, I look forward to reading your comments. Thanks.